My name is Shreya, and my teammates and I are part of Gümüş Lab at Mount Sinai School of Medicine. Today, we are excited to present to you PhosProViz, a web-based tool to generate, explore, and share interactive phosphoproteomics networks in 3D. Let's go into our motivation for building this tool. To better understand human cellular systems and how diseases influence cells and protein expression, proteomics researchers are profiling cells before and after infection, vaccination, and treatment, collecting massive datasets. Some of these studies involve protein phosphorylation interactions and perturbations. Protein phosphorylation is a vital modification for cellular processes and signaling networks. Mass spectrometry-based phosphoproteomics experiments identify and quantify thousands of phosphorylated peptides, which is very rich and complex data. This phosphoproteomic data is best represented and understood by visualizing them as networks. Typically, nodes are kinases or substrates, which are types of proteins, and edges are interactions between them. A tool that allows researchers to visualize this data in an intuitive, interactive, and unbiased way can help them better understand the immune system and how to better treat infectious diseases such as HIV or COVID-19. However, current workflows and tools to visually explore kinase substrate interactions, along with the phosphorylation sites in different states, is laborious and time-consuming. Let's talk about what tools currently exist. One tool our users commonly use is Cytoscape. Cytoscape is a 2D visualization tool where nodes and edges may be obscured or overlapped, making the network look cluttered. The user has to manually adjust parameters and thresholds for which data will appear in the network. This process can be automated, but requires programming knowledge. One specific user who had programming experience used a scripting language called Perl to threshold and color code his data. Even though the user had scripting experience and was able to automate some parts of this process, it took him several weeks to generate a visualization that fit his needs, and the process was described by him as a nightmare. We aim to develop a user-friendly, web-based tool, PhosProViz, that empowers users with even minimal computational skills to create shareable and interactive visualizations of phosphoproteomics network data. Let's talk about some of the features. Some features we'd like to highlight about PhosProViz is, first of all, and most importantly, it allows users to visualize all proteins, their connections, and phosphorylation sites simultaneously across multiple states. The tool is modular, meaning that it can be used to build more specialized interfaces with specific requirements. Some examples of tools we have built are for SARS-CoV and chronic fatigue syndrome network data. A very important feature requested by our users is that the tool operates purely in a web browser, meaning that it's very fast and easy to use without any extra configurations needed. PhosProViz also has the functionality to compare data across different states or time points. Soon, we will add animations to travel through different states and networks. Now it's time for a demo. The data import process is very simple. The tool will ask a series of questions to help you prepare your data to be visualized. The first question it's going to ask is if your network is directed or not. We're going to say yes. Next, we'll ask about the source attributes. So in other words, do we have any value that would make the kinases in the network have different colors or different sizes? We're going to say both color and size. Next, we'll be asked if our target attributes have phosphorylation sites. We'll say yes. Then we'll be asked about the color and size information for our target. We'll say we have both color and size that we want to visualize. Next, we can import information about our edges. Let's say we just have color that we want to visualize. Next, we'll be asked about our states. Does our data have different states like time points or treatments that we want to visualize? For the purposes of this demo, we'll say no, we'll just be looking at one state. Awesome. So at this point, we'll tell you how to format your data in a CSV file and how to label your columns according to the information you provided. Then you can upload your CSV file to be visualized. We'll be using COVID data for this visualization. Let's click upload. And here is the visualization. As nodes are represented in 3D, you can rotate the space, zoom into a specific node, and drag nodes around to position them. You can also take a snapshot of the current view with this button on the left. The spheres you see here are kinases and the cylinders are substrates. The substrates have phosphorylation sites, which are shown as partitions or segments on the cylinders. Log fold change values are represented as colors. Red is for upregulation and blue is for downregulation. You can change the threshold on the left and see the visualization change accordingly. You can also search for specific nodes with this query box here. And lastly, if you click on a specific node, you'll be redirected to Phosphosite Plus for more information. PhosProViz will significantly lower the barriers for researchers who want quick, intuitive, and high-quality interactive visualizations to help translate their rich data into biological insights. Hence, our tool would be useful to a wide array of professionals, including immunologists, geneticists, computational biologists, and data scientists. Thank you for watching. We'd love to hear any feedback and comments, so please reach out to us through the tool website if you'd like to chat. Thank you.